Protein Function 1, How Proteins Work. So the take-home message of this video is that proteins work through binding. And you'll see that as a big theme as you look across the vocabulary and learning objectives. Something else I want to mention here is that what we are really focusing on in this course is the biological role of proteins. And something to keep in mind as we talk about this is that biology is determined by chemistry, chemical interactions, and you'll see that quite a bit in this course as we talk about enzymatic reactions and other chemical reactions. And more specifically, chemistry is governed by rules of physics. And so while we're not focusing on those courses in particular through this course, know that that we will touch on those topics, and so it might be useful to have some chemistry resources or look at some of the earlier chapters within our textbook. For example, chapter 3 has quite a bit about enzymes. Also, this is one reason why we have these courses as part of a science degree, is to take biology, chemistry, and physics to some degree, specializing in one or the other. So let's talk about ligands. So ligand, or ligand as they're sometimes pronounced. I prefer to pronounce it as ligand, just kind of depends on the scientist. But a ligand is any substance that is bound by a protein. And some examples of substances that could be ligands are ions, a small organic molecule, and even macromolecules. And basically in this image over here, you can see this protein, and it is binding to a ligand. Note that they are a similar shape, and so this ligand has a very specific location that it can bind within this particular protein. And more specifically, that the binding is occurring through non-covalent bonds. So each of these little pink lines indicates a non-covalent bond. And notice that, at least in this example, they're spread across the surface which the ligand interacts with the protein. And so anytime we talk about binding, this is basically a interaction between these two different types of molecules. And it takes place over some length of time. So binding, what does that mean? We can talk about the length of time. And so certain ligand protein combinations will bind for a longer period of time than others. However, if this is going to be some sort of actual interaction or chemical reaction, then we at least need a length of time for the reaction to occur. A second aspect to binding is specificity. This is a vocabulary word. So specificity could range from high to low, and this is based on affinity, how much the two molecules are attracted to each other. And so basically the specificity is something that we try to depict usually in figures by shape. So showing that they have kind of complementary shapes shows that there, or at least implies that there's a high specificity. So shape matters always for proteins, but specifically for binding. And so basically physically, this ligand and protein need to be able to get in close contact with one another. And that's to allow those non-covalent bonds or non-covalent interactions to form. And so sometimes a ligand and protein binding is compared to a hand and a glove. So certainly your hand, you can put on all sorts of different sizes of gloves, but certain gloves will fit more tightly, and then they maybe will spend more time on your hand because you can use them more than others. So if you had a glove that was way too large, wouldn't be very useful, and you might just choose to take it off. or it might fall off if it was even too big. And so that's the case with ligands and proteins as well. You can also see in this figure that they've got kind of a space-filling model, checking out the ligand again in yellow and the protein in green. And this is showing just how tight that these two are fitting together. And so basically, again, that tightness is important because that allows the non-covalent bonds to form. Distance matters for non-covalent interactions. Okay, let's talk a little bit about ligand binding affinity. This is due to 4 interactions, the 4 non-covalent interactions. So take a minute and remember what those are. They are hydrogen bonds, electrostatic interactions, van der Waals attractions, and hydrophobic interactions. And basically, any ligand binding affinity requires many interactions. So just one of those interactions, because all of those interactions are individually weak, is not enough to produce any sort of binding for any substantial length of time or across any physical distance. So basically, it requires many interactions for this affinity. Let's take a look at this figure now to talk about molecules and a couple different scenarios. So basically for this figure, we're going to talk about a particular molecule, molecule A, this kind of light brown, orangey looking thing. And so basically molecule A can interact potentially with 3 different molecules, B, C, and D. And so basically molecules don't have a tractor beam where they can, and they can't see a molecule that they might fit well with. They are blind, and so basically molecules are just floating around in 3-dimensional space in the cell. And so basically they're just trying out different fits across whatever they bump into. And whatever they fit best with, or have the strongest interaction or strongest affinity, then they're going to spend more time in that particular interaction. And so basically you can see here, so molecule A can bump into molecule B, and it does form some of these red lines, these non-covalent interactions. But notice that that kind of falls apart pretty quickly. A and C, if we count up the number of non-covalent interactions, they have one more non-covalent interaction, but still that doesn't really add up to much. And so they're going to also kind of go on to their own separate next step, try to bump into something else. And in this case, if A were to bump into this molecule D, notice that there are a lot more non-covalent interactions which take place. And so then this particular interaction would be stronger, and basically it would last for a longer period of time. So some take-home messages from this figure are, keep in mind these red lines each indicate non-covalent interactions. And that the more of these non-covalent interactions that are present, the longer 2 molecules will associate with one another or bind to one another. And then this is a higher affinity between the 2 of them. And again, this length of time is crucial for allowing a chemical reaction to take place. So let's talk about the binding site next. So we're going to zoom in to look at particular locations where a ligand and a protein will physically connect with each other, and basically where those non-covalent interactions are forming. So binding site, this is a region of a protein that associates with a ligand. And the implication that this is over some substantial length of time. And a lot of times this is going to be sort of cavity or opening, and this is formed by the shape of the protein. And as you already know, the shape of the protein is due to the amino acid R groups. And certainly shape of protein can also be determined by the backbone. In this case, most of these cavities or openings are going to be more complex than just an alpha helix or beta sheet. And so in this case, we're looking at the R group interactions. This is figure 4-32 is pretty similar to that. This is an older version or newer version from a different book. And basically, let's take a look at that. So this particular figure, here we see the chain of amino acids in the polypeptide chain, and then the R groups are shown in these different colors, and those are basically forming not a whole lot of discrete shape here since it's an unfolded protein. As protein folding takes place, then notice that all of these amino acid side chains start to orient each other and form some sort of stable shape, and then this little divot here, or cavity, that's the binding site where something can actually fit in place. And so protein has to be folded to have this three-dimensional binding site. Notice also that this color coding that we see on this part of the figure is going to hold true. So this little orange spot here refers to this R group over here, yellow, and so on. So just a way to kind of help you look through the figure. And so now we're going to zoom in on this part, and so we'll zoom in over here to check out those R groups and some of the non-covalent interactions. So basically what you can see here is that we've got some hydrogen bonds, these little pink lines, and then we've got this green thing here in the middle, this chemical structure. This is cyclic AMP, also abbreviated little c, big capital AMP, and so that is a modified nucleotide. It's actually a derivative of ATP, and it's used in signaling cascades. We're going to hear more about cyclic AMP later. For now, this is just showing us a ligand and how it can interact through these number of non-covalent interactions, specifically hydrogen bonds, to hold this ligand in place. And something else to notice is so certain amino acids which are lining the binding site, those are actively forming interactions with this ligand. But know that the other amino acids, so ones that are not shown in detail, like over here in these little dash lines areas, those are forming a scaffold, so basically kind of a support structure for the rest of these to hold in shape. So the rest of the protein is still important, it's just not shown in this part of the figure. And again, we get to use this word scaffold just to say that that three-dimensional structure is also relevant. So I'll make a little note about that. So maybe I'll use the pen. So amino acids that are not involved in the shape of the binding site presumably have a role in the scaffold for the overall shape of the protein. And so it's only through experiments that we could find out, well, which parts of the protein are really crucial, which parts are the domains, and those are some things that molecular biologists are often very interested in, as cutting up proteins and then determining what's kind of the minimum part that's necessary for a particular interaction to take place. And so eventually when we look at some primary literature data, we'll look at some of those modified proteins and learn about how they do that.